Hello, and welcome back to educator.com. Today we're going to talk about DNA mutations and repair. Now, as an overview, first thing we have to talk about is what is the difference between DNA damage and a DNA mutation. Once we understand that difference, we can talk about the various types of DNA mutations that we will come across. And when we come across mutations, hopefully we don't even get that far and we can go through DNA repair. We'll talk about several types of DNA repair processes, and we will finish up getting ready to move on to the next unit. So first of all, what's the difference between DNA damage and DNA mutation? Well, DNA damage is just an alteration of the chemical structure of DNA. And that can be a break in the DNA backbone, we could have a loss of a nucleotide base, or we could have some sort of chemical alteration of that nucleotide. Now, DNA mutation is a permanent change of that nucleotide sequence. So the mutation will result from DNA damage that is not repaired properly. Right? And it can also result from our replication errors. It can be exogenous or endogenous types of damages that aren't repaired. Or we can have what are called indels. And indels stands for insertions or deletions. Okay? And these are often a result of transposable genetic elements, which we call transposons. Now, some of the classes of the DNA mutations, the two main ones that we're going to see are either spontaneous mutations or induced mutations. All right, our spontaneous mutations are our endogenous damage. All right, just as a result of molecular decay. So these are mutations that are due to error-prone replication bypass of naturally occurring DNA damage. And usually this is from error-prone translesion synthesis, which is a type of DNA repair mechanism that we will talk about later in this unit. These errors are usually introduced during DNA repair. Now, we also have induced mutations. Okay, and these are going to be exogenous damage. So this is going to be DNA damage caused by mutagens, whether that be chemicals or radiation. All right. And what we're eventually going to end up with, no matter which one we're looking at, is if we have damage and we don't have proper repair, we're going to end up with some sort of pathology, so some sort of possible disease symptoms. And if we see here, a disease cell is when we have more damage than repair going on. A healthy cell will be when we have just as much repair going on as we have damage, so that it's almost like nothing ever happened. Now, some of our spontaneous mutations that we can come across are tautomerism, and that is basically a cause of why certain bases will mispair with other bases. Remember, our A's should always double hydrogen bond to T's, and our G's should always triple hydrogen bond with C's. Okay, so tautomerism is the repositioning of a hydrogen atom, which alters the hydrogen bonding pattern, resulting in incorrect base pairing. So this could be, an A maybe pairing with a G or a C, or, a G pairing with maybe an A or a T instead of that correct C. All right. Now, depurination is the loss of a purine base, forming what's called an apurinic site, and all that means is it's a site that doesn't have the purine base, so it doesn't have the nucleotide there. The DNA backbone is still there. The sugar, the deoxyribose is still there. We just don't have the proper base. Okay. Another type is what's called deamination. And that's a loss of an amine group. Remember, our amine groups are NH2, okay, attached to the alkyl group. And so we lose the amine group, replacing that with a ketone group. So deamination of cytosine will lead to a uracil, because cytosine and uracil only differ by that amine group. Okay, if you deaminate adenine, you can end up with hypoxanthine. And if you deaminate your 5-methylcytosine, that will give you a T. And let's just look at this one really quickly. If we have a cytosine, right? Cytosine should triple hydrogen bond with our guanine up there. But if it's changed to a T, right? If it's changed to a T, what are we going to do? Now that T is going to hydrogen bond with an A instead of the original G. So that can affect what type of mutations we see in future generations of the DNA. One of the last type of spontaneous mutations we're going to talk about is called slippage or slip strand mispairing. And what this is that we have denaturation of the new strand from our template during replication, and that's followed by the renaturation in a different spot. And this can cause deletions. Okay, we're going to talk a little bit about this slippage in a few slides, in which case I'll draw out an example so that we can see what is actually going on here. All right, so some of our induced mutations and their causes. So chemicals, right? Chemicals are a type or what can induce mutations. And some of the chemicals that can do that are such things as base analogs, alkylating agents, DNA intercalating agents, DNA cross-linkers, as well as oxidative damage. Now, our alkylating agents can mutate both replicating and non-replicating DNA. All right, whereas base analogs can only mutate DNA that is undergoing replication. All right, so that's a difference for those. Our intercalating agents are molecules that get in between two strands of DNA, and I'll show you an example of that when we talk about a tobacco smoke related mutation. And crosslinkers just covalently crosslink two pieces of DNA together. And oxidative damage is something that we see all throughout. And oxygen is just a molecule that can be good or bad depending on what you're looking at, right? We need oxygen to breathe, but excess oxygen, especially reactive oxygen species, can be very detrimental and very reactive, causing a lot of damage. Now, another type of induced mutation, or what can cause induced mutations, is radiation. And we have non-ionizing radiation as well as ionizing radiation. Now, an example of IR would be X-rays, okay, gamma rays. And so IR produces really, really highly reactive free radicals that can break bonds in the DNA backbone. And if we break the DNA backbone, that can be extremely detrimental, especially when trying to replicate new DNA or transcribe through portions of broken DNA. So some examples of our IR, as I mentioned before, we have our gamma rays. We also have our X-rays. Remember, gamma rays are what is found in the Hulk, right? That turned Bruce Banner into the Hulk. X-rays, if you're going and get a scan trying to see through your body, looking at your bones, as well as you'll encounter X-rays when you go flying. Okay, they're in a lot of places. Cosmic rays are what is in outer space. Okay, and it's kind of everywhere. It doesn't reach through the atmosphere very well, but they are quite abundant. Okay, and some examples of our non-ionizing radiation are UV light, visible light, infrared, microwaves, and radio waves. So our ionizing radiation has a shorter wavelength and a higher energy. Okay, so shorter wavelength, higher energy. Okay. Whereas our non-ionizing radiation has a longer wavelength and therefore lower energy. Okay. Now, I said that I'd mention what a DNA intercalator is. And what we have here, just down here to skip down to the second bullet point, is what we're looking at on the right side. This is a covalent adduct between DNA and benzopyrene. Right? And this is the major mutagen found in tobacco smoke. Now, what a DNA intercalator is, okay, so benzopyrenes are DNA intercalators. And what a DNA intercalator is, it is something that binds in between the two DNA strands and kind of pushes it apart. So if we see here, the benzopyrene is right here, okay? And it is affecting the DNA strand in its nice overall helical shape, and it is really pushing the two DNA strands apart and kind of the base interactions up. So not only are we pushing outward, but we're kind of pushing the base stacking interactions down. And it's actually affecting, if we look at right here, okay, these two nucleotides on opposite strands are not able to bind right there properly. Okay, or it may affect how many hydrogen bonds can be made. So our benzopyrenes are categorized as pollutants as well as carcinogens, right? Something that can cause cancer. They are naturally emitted by forest fires and volcanic eruptions. And they're also found in coal tar, cigarette smoke, as I'm talking about right here, and wood smoke. So this doesn't sound like something found in a product that we want to be putting inside of our bodies, all right? So another example of a DNA intercalator is ethidium bromide, which is something that we use in the laboratory to help us visualize DNA. However, it is mutagenic, it's carcinogenic, it can cause cancer because it will get in between pieces of DNA and cause alterations, and this can affect the DNA in future generations if this causes a problem in the proper replication or transcription of the DNA. So one of the DNA mutations that I want to talk about is caused by UV light. Okay, so UV damage. Remember, UV light is a type of non-ionizing radiation. Okay, and so oxidative damage occurs from UVA exposure. All right, and one of the main things we have occurring from this is what's called a thymidine dimer. And what that is, it is a covalent bond between adjacent thymines on DNA, on the same strand of DNA, which forms a cyclobutyl or cyclobutane ring within the T's, which does not allow them to properly base pair with A's on the opposite strand. So this would be normal. Okay, and this right here would be our cyclobutyl, right? So we have T's binding together here instead of across the way to the A's, all right? And this is highly mutagenic, and this can cause big time problems because you can't replicate or transcribe through these places. So we have to repair them. So let's talk about this real quickly. All right, so our UVB rays, okay, they're shorter wavelength, which means they usually have higher energy, right? Shorter wavelength, higher energy. They usually don't penetrate past the epidermis, right? So it's the outer skin layer. But UVB rays are the main cause of sunburns and skin cancers, right? And UVB rays account for about 5% of the total UV light that reaches Earth, okay? Now, UVA rays are longer and therefore less, okay, lower energy. But they will penetrate through the epidermis down deep into the dermis, so below that top layer, okay? Now, UVA ray exposure may lead to premature skin aging, wrinkling, and immune suppression. And UVA rays are about, okay, 95% of the total UV light that reaches Earth. And then we also have UVC rays. Okay, UVA is the dominant tanning ray. Okay, so UVB, okay, that's going to sunburn you. UVA, more likely to tan you. All right. Okay. Now that we know what UVA and UVB are, let's talk about what we want to know regarding this, right? So we want to know, how does this affect us when we go out into the sun? And how can we protect ourselves, right? So we all know about sunblock, and we've probably all seen the SPF factor, right? The SPF 10, 15, 30, right? 50, SPF 90, right? So what does that actually mean? Well, SPF is what's called sun protection factor, okay? And what that tells us is it says how long it will take UVB rays to redden treated skin compared with untreated skin. So for example, if we take a sunblock that says it's SPF 15, what that means is that if we put it on, say, our left arm and not our right arm, it will take our left arm, okay, the treated skin, 15 times longer, okay, to get to the same redness as this arm, okay? So if I put this arm out for an hour in the sun, I'll get some sort of redness, okay? Well, treated with SPF 15, this arm would take 15 hours of the same sun exposure to get to the same redness level. All right, so this is a way that we can protect ourselves from these harmful rays, because remember, our UVB, not only the main cause of sunburns, but also the main cause of cancers. And just as another aside, those fake tanning booths, they're not much safer than the sun, right? You still would need to use your SPFs, because actually fake tanning increases your risk for skin cancer by at least around twofold. Right. So if we don't repair our DNA, we will lead to mutations. And here are a list of many separate mutations that can be found from originally having a mutation in DNA, and then right here we're showing the codon table, so the mutation in the DNA gets transferred to the RNA, and that mutation from the RNA, right, from DNA to RNA is transcription, and then the mutation transcription to RNA to protein translation will give us a problem in a functional protein, or maybe a problem making that protein non-functional anymore. So what we're saying here is that DNA errors not repaired make DNA mutations, and DNA mutations can lead to disease. So we want to be able to figure out a way to stop that. So let's look at the possible repair mechanisms. All right, so we are going to talk about all of these today, and that we have base excision repair, nucleotide excision repair, recombinational repair, so homologous recombination, non-homologous end-joining, as well as one that's not listed on here, which is called alternative end-joining. We will start with mismatch repair, and we will briefly mention direct reversal. So that's not so much a repair mechanism as it is exactly what it says. It's a direct reversal, let's say, before a repair mechanism would have to come in. Okay. So mismatch repair, it says up here, mainly we are fixing replication errors. All right, so mismatch repairs, we're going to fix our errors in replication. All right, and so the normal error rate of replication is 10 to the negative 5. So that's 1 every 100,000. 1 every 100,000 bases. All right, that's not, I mean, that's great, but remember, we have over 3 billion bases in a haploid genome. So we need to get better than that, or else there's too many errors that can lead to mutations, and maybe it can lead to apoptosis or death of those cells. If you have too many cells dying, you have, as the organism will eventually die. Okay, well, proofreading mechanisms increase that, right, to 1 in 10 million, right? That's great. Still not good enough. Okay. 1, 2, 3, 4, 5, 6, 7. Okay. Well, MMR, right? MMR, mismatch repair, can bring us to 1 times 10 to the negative 10, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, that is 1 in 10 billion bases. So we only have a 3 billion base pair genome. So this is saying, hey, by way of this, we shouldn't have any errors through an entire replication cycle. And that is a great thing to see. All right, and that's what we want. So MMR has to repair our mismatches, right? Otherwise, they become mutations, and mutations can lead to disease. So how does MMR repair our mismatches? So first thing, if we look down here, okay, on the left side, this is how eukaryotes utilize mismatch replication. On the right is how E. coli utilizes mismatch replication, or mismatch repair, sorry, MMR. And then we have the majority of bacteria is what's using the middle. So I'm gonna talk about the eukaryotic, right? Since we are eukaryotes as humans. So first thing, you have to recognize the mismatch. So over here we see a GT, right? That's not a proper mismatch. We should have either an AT or a GC. So the mismatch is recognized by mut S alpha. Okay, that's a protein complex, or a protein. Okay, then we have an incision in the DNA backbone, and that's done by mut L alpha. Okay, next we remove a stretch of the DNA, including that mismatch, and that is done by XO1. Okay, it's a nuclease, as well as RPA being involved. Remember, that's going to help bind to the single-stranded DNA. Then, once we remove that, we can synthesize new DNA. So we have DNA polymerase delta coming in, synthesizing new DNA, and finally, DNA ligase ligating the gap or the nick in the DNA backbone. Okay, and it's the same thing happening in bacteria as well as E. coli. It's just different protein names. Okay. Now, an important thing to see is, how do we recognize a mismatch in the first place? Okay, and how we see that mismatch is due to the fact that this is somewhat bulky. Okay, this mismatch is bulky, and that is how this original protein, mut S alpha, if we're talking about eukaryotes, finds that mismatch. Okay, but just as important of a question is, how can we tell which of the two strands has the error? Okay, which one is actually what it's supposed to be? Okay, so is the G the error, or is the T the error? Well, how you figure out which one has the error is you have to recognize which one is the parental strand versus which one is the newly synthesized nascent strand, because the parental strand will have what the proper sequence should be, and the error will be in the newly synthesized strand. Okay, so in E. coli, how you recognize a parental strand is that mut H will bind to methylated sites, okay, on the DNA, and it will nick the non-methylated strand. So in E. coli, they find these sites that are called GATC, okay? And this adenine will have a methyl group added to it, all right? And this occurs in E. coli, and it's as a defense mechanism so that any non-methylated DNA that is in the cell, it sees as foreign, so it will cut it up. So say say a virus is trying to kill the bacteria. So the virus latches on, expels its DNA in there. Well, the E. coli can utilize a defense mechanism that breaks up any DNA that is non-methylated. Well, you can't methylate DNA right away after synthesis, so there is a short period of time where the newly synthesized DNA is unmethylated. So that's how E. coli can recognize the parental strand versus the newly synthesized strand. And so once it's recognized, we have RecJ, which is a 5' to 3' exonuclease, or EXO1, which is a 3' to 5' nuclease, it'll bind and remove that stretch of DNA, including the mismatch, from only the non-methylated strand. It will not do this on the methylated strand. So this will only be removed from the newly synthesized strand, leaving the correct parental strand sequence there. And then when you go through the resynthesis process, you will hopefully this time synthesize across the proper parental strand, giving you the, once again, correct, let's say, AT base pair. Okay, instead of what it was showing was a GT. All right. Now, eukaryotes and many prokaryotes do not methylate their strands. So this one is a little bit harder to think about. But now our parental strands are unbroken, okay, during replication. We know that. And our nascent lagging strands, as we've already talked about, has nicks, right? Because of the Okazaki fragments, right? The polarity of how DNA polymerase needs to synthesize 5' to 3' leaves us with a lot of nicks. So that's how eukaryotes and a lot of prokaryotes can recognize which strand is parental versus which strand is newly synthesized nascent. So we have EXO1, a 3' to 5' exonuclease, binding and removing that stretch of DNA, including the mismatch, from the nascent strand only. All right, and then we have our DNA pol delta coming back in, resynthesizing, hopefully this time, the correct nucleotide, and then we have DNA ligase sealing the gap. So I had said earlier that we talked about slippage, and I would draw this out so that we can understand a little more. So mismatch repair can also repair loops in the nascent strand due to what's called slippage. So if we have a piece of DNA, let's call that. All right, so this would be a GTC, GTC, GTC. Okay, so if we have repeat sequences, all right, this can be problematic during replication because what can happen is that we can have what I said is called slippage. So CAG, okay. And during slippage, we can have this, we can have some denaturation to where we have it coming off of the proper sequence. Okay. And what we have, okay. All right, and we can draw this like this so it looks easier. GTC. Okay, and this one is actually binding to this. Okay, so what we have is, if this happens, if we slip this loop upward, but we're still binding properly, say here, right? All right, this is all hydrogen bound. We end up with an increase, okay, in our DNA. So we end up with what's called excess repeats. Okay, so this, if we write this over here, so this is going to be an expansion, and in this case it's a triplet expansion because there are 3 bases, right? So these are going to end up being added in when they weren't before. So let's draw this again. We have CAG. Okay, and then these have gotten added. Okay, so what's going to end up happening is that we're going to end up having to fill in this excess. Okay, so GTC, GTC, GTC. So what would happen is we can have, this is our mutant DNA. Okay, so this has all been added. Okay, and this is problematic because as a triplet repeat, especially when we're talking about this one right here, this is what we call a polyglutamine repeat. The reason being that when this CAG, okay, when it codes, this will code for our glutamine when we're talking about turning it into mRNA and then finally into a protein. So this will code for glutamine. And there are several diseases that are associated with polyglutamine expansion. A couple of those are Huntington's disease, as well as spinocerebellar ataxia. Actually, 8 different types of that. Okay, and if we're talking about just triplet expansions, not necessarily the polyglutamine ones, we also have diseases such as Fragile X syndrome and Friedreich's ataxia as well. And so these can be extremely detrimental, and what we have here is the more repeats that we see, the more likely you are to go through slippage the next time you go through DNA replication. So we have mismatch repair hopefully being able to catch this, remove these, right? Hopefully we can remove these so that we don't have the expansion and we just can go back to our original DNA sequence there so that we don't have the expansion and we don't lead to disease. So MMR plays a huge part in that. So I told you we would very briefly mention damage reversal. Okay, and what damage reversal is, it is exactly what it says. So you're reversing the damage. It's before you have to go through a true DNA repair mechanism like mismatch repair or the other ones that we're going to talk later. So an example of direct reversal or damage reversal is the direct reversal of pyrimidine dimers. Okay, remember our pyrimidines are our C's, our U's, and our T's. If we're talking about DNA, hopefully we don't have any uracil in our DNA, right? So if we're talking about DNA, hopefully we're just saying cytosine and thymine. Okay, and our pyrimidine dimers, often as we talked about before, caused by the UVA light, the thymidine dimers. Okay, so direct reversal is catalyzed by, in this specific case, DNA photolyase. That's an enzyme that will use energy from visible light to break the cyclobutyl ring formed between thymidines. And if you break that if we look here, okay, here's our DNA. Our thymidines are now bonded together instead of to the adenines across the way. Well, if we can get photolyase to come in there, okay, photolyase can break that bond, okay, and then help you make the new bond or the proper bonds across the way between our adenines and thymines. All right, so that's an example of damage reversal. All right, so on to another type of repair, and this is a very important. All of them are very important ones, but this is one that we're going to use for small and non-bulky lesions. However, we see these a decent amount. So basic excision repair, otherwise known as BER. Okay? Uses specific DNA glycosylases to remove abnormal bases that are due to oxidation, alkylation, or deamination. Okay? And so these types of mutations are often caused by IR, right? Ionizing radiation, alkylating agent, oxygen radicals, or spontaneous hydrolysis. Hydrolysis, remember, is just the breaking of something. And so what we have is we see a damaged base. Okay, the glycosylase comes in there, recognizes it, recruits a nuclease, an endonuclease, to cut that piece out, and then we'll just resynthesize the correct base and religate the gap. So we have DNA pol beta, okay, being involved in short patch repair, okay, if we look on the right side, and we have DNA pol delta and epsilon involved in long patch repair for DNA synthesis. Okay, and all that means is the length of the repair process. Okay, few bases, several bases. So here's an example. So if we have the spontaneous deamination of cytosine, so let's draw this out. Here's a DNA sequence. A, T, C, G, A, C, T, G. Okay, so that's going to be T, A, G, C, T, G, A, C. Okay, so if we have spontaneous deamination, okay, and that is when we have our amine group coming off, right? Okay, so what that leads us to is A, T, C, G, A, C, T, G. Okay, and then it is TAG. And let's say this C was the one that got deaminated. So now, remember, I told you before that C can be deaminated to a U. Okay, so this is T, G, A, C. Oh. Okay, so what we have, once we see that spontaneous deamination, we have what's called uracil glycosylase coming in and cutting this base from the backbone, leaving what's called an A basic site. So if we continue on this way, right, so we have uracil glycosylase. So we have, here we go, A, T, C, G, A, C, T, G. We have T, A, G, T, G, A, G. Okay, so right here, we no longer have the base because it's been removed by uracil glycosylase. Okay, so it is cutting at the place of where the uracil is attached to the deoxyribose. Okay, so it's hydrolyzing that bond. Okay, so the next thing that will happen is that we have AP endonuclease nicking the phosphodiester backbone, which is 5' to the A basic site. Okay, so A, T, C, G, A, C, T, G. T, A, G. T, G, A, C. Okay. Okay, so we have nicked that backbone, okay, 5' to that A basic site. And an A basic site just means it's a site without any base. So it doesn't, A basic means it could be a purine or it could be a pyrimidine. But here is an A basic site right here that we're looking at, okay? And here we have the nick in the backbone due to the action of AP endonuclease, okay? So the next step, we have deoxyribose phosphate lyase removing the deoxyribose from the backbone. So let's see this. Once again, I'll write out what we were starting with. Okay, so we had A, T, C, G, A, C, T, G, T, A, G, T, G, A, C. Okay, so remember guys, this is just right after AP endonuclease has worked. All right, now we have deoxyribose phosphate lyase. Okay, and what we have here is we're going to remove the deoxyribose from the backbone. So what we have is 5' ATC GACTG. We have TAG TGAC. Okay. Now, remember, AP endonuclease over here just made a nick, but it didn't remove the actual deoxyribose. That is where deoxyribose phosphate lyase comes in. It removes the entire thing, right? So now we have a full break in that backbone right there. We are missing the sugar. Okay, so the final thing that happens is we have DNA polymerase and DNA ligase coming in. Oop. Okay. Okay, we're going to add a new deoxy-CTP, removing a pyrophosphate, okay, so that it enters as a monophosphate. And we end up with, all right, okay, we end up with the repaired DNA. ATCGACTG TAG. Okay, and then the C, as well as the new deoxyribose, has been added, and we're back to normal what we had. Okay, so this is the full repair using base excision repair. All right, good. So as I said, DNA polymerase and DNA ligase bring in the correct cytosine nucleotide, and they seal the nick in that phosphate backbone. And it's good as new, like it never happened. Okay. Another type of repair now, okay, to switch a little, is nucleotide excision repair, otherwise known as NER. Okay. And NER recognizes a wide range of our DNA damage, including our damage bases, the pyrimidine dimers that we've already talked about, as well as bulky adducts, and those are just big cross-link molecules. Now, defects in NER can result in disease. Okay. Now, a couple diseases to mention is XP, xeroderma pigmentosum, and that often presents with photosensitivity. So you're very sensitive to light. Okay, can lead to skin lesions and cancer. And down here is a patient with XP. All right, you can see that they have a lot of skin pigmentation, and this is due to the fact that we are accumulating these problems, okay, from UV light, these damage bases, dimers, all these things from UV light all the time, and we need to figure out a way to repair them. Well, when we have a problem in a main repair process, that builds up over time. And actually, people with XP can't be in sunlight for very long by any means. And when they do go outside, they have to go out like in fully clothed. It almost looks like a beekeeper's uniform, in that they have a hat with a face shield, and their body should be covered so that you don't end up with the accumulation of all these mutations. Okay? Another defect of a defect of NER can produce another disease called Cockayne syndrome. Okay, and that also presents with photosensitivity, as well as growth failure and neurodegeneration. Now, let's talk about nucleotide excision repair, and we'll talk about it being in E. coli first, because the mammalian version is, while it's very similar, it is much more complex and has 25 or more proteins involved. So same concept, just a lot more complicated. So let's talk about E. coli. So first thing that happens, UVRA and UVRB proteins scan the DNA for distortions. So this is number one right here. UVRB will melt open the helix next, okay? And UVRC will cleave on both sides of the lesion, about 12 to 13 base pairs. Okay, so this is 3. Next, UVRD helicase will remove the lesion. Okay, so this is 4. And then DNA polymerase 1 will fill in the gap, and then DNA ligase will seal that nick. Okay, so this is also as I've written it. Okay, number 5. Okay, so it's a fairly simple concept, but a very important part. Okay, very important for the proper maintenance of the genome. Just so, I have it listed later, but just so we know, UVRA and UVRB in humans, the protein that performs the same function is XPC. UVRB, the human analog would be XPA, XPD. Ooh. UVRC, that is similar to XPG and ERCC1 XPF complex. And UVRD, or doesn't really have as great of an analog. And we have, instead of a 12 to 13 base pair gap in bacteria, in humans or eukaryotes, we have about a 24 to 32 base pair lesion. Okay. And as I said, many more proteins involved. So there's two different types of NER that we can accomplish or complete in eukaryotes. First is global genome nucleotide excision repair, and then the second one is transcription coupled NER. And the difference is that global genome NER can repair damage in both the transcribed as well as non-transcribed DNA strands in both active and inactive genes throughout the genome. Transcription coupled NER can only be performed on actively transcribed genes. Okay? Only. Okay? Now, transcription coupled NER, so tcNER, rescues a stalled RNA polymerase at whatever this lesion is. Okay? And what we have is transcription factor 2H, TF2H, participating in general transcription as well as transcription coupled NER. So it's already there. So it participates via its XPA and XPD subunits. Okay, so the helicase. And this is important to help get RNA polymerase 2 through this lesion and continue on through transcription. Otherwise, we can have problems and eventually lead to the cell undergoing apoptosis. Okay, so I talked about apoptosis. Remember, apoptosis is the programmed killing of cells. Okay, and so we talk about that being a good thing sometimes, right? Because if you don't go through apoptosis, you can end up having unchecked cell proliferation, which we know leading to cancer. So let's compare MMR and NER. So it doesn't matter which one we are looking at, they all go through a similar series of steps. So first things first. Okay? There's a surveillance of the DNA damaged lesion. So you need to recognize that there's a problem. Okay? Then you induce a cell cycle checkpoint. So you don't want the cell to continue on like it's normal. You need to repair that lesion first. Okay? So you activate those repair networks. Okay, then you'll go through DNA repair and you will keep the cell cycle in an arrest. Okay, you don't let it go on any further. And you can go one of two ways at that point. Either you have figured out that the repair cannot be done and you go and finish with apoptosis. So you kill the cell. Okay, say, okay, it's beyond repair. We're just going to kill the cell and we'll start over. Or we go through and we proofread, we repair, we do everything, and we think we're on the right track. Okay, so we're going down this way. From there, okay, we have two options once again. Everything is okay. Everything's okay, so we go on to the next process of whatever we're supposed to do in the cell, right? So from replication, right, we could go S phase, then we can start going through mitosis. Or if we still see that there's a problem, we can once again have a point where we can go through apoptosis. Too much has happened. We didn't fix it correctly, so we're going to kill that cell. We'll start fresh. Okay. So both of these pathways are very similar in that effect of we look for the damage, we induce a cell cycle checkpoint, so we stall it. We repair, and then we check to see if we actually repaired it properly, in which case we can continue on, or if we didn't repair it properly, we kill the cell off. Okay. All right. So one of the last things that I'm going to talk about is what's called translesion synthesis. And translesion synthesis really isn't a repair process. I know this is in the DNA repair section, but we need to talk about it. And by the way, translesion synthesis is also abbreviated TLS. And it's not a repair process, it's more of a damage tolerance mechanism. Okay, so this can be induced by our cell cycle checkpoints. If we're talking about eukaryotes or bacteria, they have an SOS response. It's basically the same thing. And this TLS allows replication past these DNA lesions by polymerase switching. So you may find, let's say example, right, in AT, right? It's usually going to be a little more involved than that, but it should be in AT, right? But we see an AG. Okay, and maybe we see a bunch of these. Okay, and that's problematic. It stops synthesis, replication, all that stuff, right? So we can't go through there. We can't figure out a way to get past it. So we just put in another, I'll actually give you a better one. Well, let's say the pyrimidine dimers, right? So these are attached to each other instead of across the way to A's. So we can't get through there. So instead of trying to synthesize through there, we just throw in another polymerase that doesn't care, right? It doesn't care what the correct nucleotide should be. It just throws stuff in to get past the gap. Okay. So how it does this is it goes through polymerase switching, and it uses low fidelity or very error prone polymerases. And these polymerases will incorporate bases that likely don't match the proper base pairing to the template strand. Okay. And these error prone polymerases are usually from the Y family of polymerases. And if we're talking about E. coli, we have polymerase 4 and 5. And in eukaryotes, we have pol eta, pol iota, and pol kappa. A cool thing about pol eta is that it automatically inserts two adenines across from a thymidine dimer. So when we see this thymidine dimer, okay, and we need to synthesize this new strand down here, right? Right, well, we can't utilize this TT as a nice template. Okay, so what it does is this pol eta says, oh, I see a TT, all right, whatever. I'm just throwing two A's in there and continuing on my synthesis. All right, so luckily we have kind of a fail-safe for that so that we can get through this process. Okay, but it's error-prone. It doesn't necessarily know that those are two T's. Okay, so the steps of TLS, first the lesion's encountered and we stall the replication fork. Secondly, we have the PCNA, right, the sliding clamp on our replicative polymerase gets ubiquitinated. Okay, ubiquitin is just a very small protein, okay, and it can be added to our sliding clamp, and that will cause the sliding clamp to release from DNA. Now, the sliding clamp and the replicative polymerase, okay? Now we can switch to a translesion synthesis polymerase that bypasses the lesion regardless of what that sequence is, right? So it's, you know, it could be T, T, A, C, G, T, C, A, right? So this is what, right? 5 prime, right? So we're trying to synthesize this way. Okay, and let's say we find the lesion being around this area. Okay, so this is fine, right? So we have a T, we have a T, we have a G, we have an A. We run into a lesion. Okay, so the replicative polymerase falls off. Okay, let's call it epsilon just for sake. Okay, and now we get a translesion synthesis polymerase coming on. Let's say pol kappa, just say. Okay, now pol kappa comes on, and it's just going to start polymerizing. Okay, maybe it gets a couple right. Okay, but then maybe it just gets ones it doesn't care, right? It's just trying to get through that lesion. Okay, and we'll say the lesion stops here. At which case, we have the TLS, right, coming off, and we have our replicative polymerase coming back on and synthesizing with high fidelity again. So these right here, this area might not exactly match properly, but it is more useful for the cell to be able to have a few mutations and get through to be able to finish all replication than for the cell to go through apoptosis. Now, yes, this could end up causing cancer, but it might also end up leading to evolution, as can be seen throughout many, many generations so far. All right, so the last type of repair I'm going to talk about is recombinational repair. And this is going to give you guys a nice overview and preview for what we're going to do in the next unit. All right, so recombinational repair repairs our double-strand breaks, okay? And these double-strand breaks are caused by ionizing radiation, okay? And if we remember right, our ionizing radiation are our cosmic rays, our gamma rays, our X-rays, right? And as I mentioned before, you can get X-rays from imaging, from radiation therapy, from flying, all kinds of ways. We have our double-strand breaks being repaired via three main mechanisms, okay? We have NHEJ, non-homologous end joining. We have alternative end joining, which we can call alt-EJ. And then we have homologous recombination, which we can call HR. Okay, we're going to talk about these, especially homologous recombination, we're going to talk about in depth next time. Okay. So, double-strand breaks form pretty rarely, but they can be catastrophic to the cell if they're not repaired. So, on average, we have about 10 double-strand breaks per cell per day occurring from replication alone. Okay, and not to mention all the other things, right? Our X-rays, you know, the exogenous stuff. All right, so if we don't repair these double-strand breaks, we can lose pieces of chromosomes. Okay, and that can be extremely detrimental, and hopefully that could lead to, let's say, if unrepaired, just apoptosis, right? You have cell death. If not repaired and doesn't go under apoptosis, you can likely form some sorts of cancers. Okay, so the mechanisms of double-strand break repair are the three that I mentioned before. So, non-homologous end joining. Okay, that's our fastest one. Takes a little less than an hour, but it's really error-prone. Okay, and when is it usually occurring? In the G1 phase or in the G0 phase. So, the senescent cells, the non-dividing cells. We have alt-EJ, right? Alternative end joining. That's used very rarely, and it's really slow, about a few hours long, and it is also error prone. Then we have the preferred mechanism, okay, homologous recombination. It is the slowest, it's about 8 to 10 hours, but it is fairly error free. Okay, and this is usually occurring in S phase or G2. So this is in S phase when we're having a lot of hopefully not mutations, but errors, okay, DNA damage. Now, non-homologous end joining. So this does not require homology to repair the double-strand break. So what it does, we have a protein complex, okay, a heterodimer, Cu7080, binding double-stranded ends and recruiting a kinase called DNA PKCS. So DNA protein kinase, CS, catalytic subunit. Okay. We then have Artemis in exo and endonuclease, removing mismatched nucleotides via resection, so cutting away the ends. We then finally have synthesis by pol mu and pol lambda, and then we ligate the ends via DNA ligase 4. Now this will result in point mutations and small deletions, but the double-strand break is repaired. So what NHEJ would look like, just very simply, Okay, we have our Cu7080 binding. Okay, and then you recruit the kinase, Artemis. Okay, it's remove that piece, then we can get proper synthesis, right? Okay, good. Alt-EJ, alternative end joining, uses microhomology to rejoin the two ends of the double-strand break. And what that means, microhomology, is it finds some sort of match, a minimum of 4 base pairs long, between the two strands. So, how that would look. Okay. Oh, let's just write that in there. Okay. So they find this, there's a double strand break, right? Which is seen here. But then you search down each individual piece, right, to where you can find a microhomology of at least 4 bases long. Okay, so we resect the ends using the MRN complex and CTIP, and you find that microhomology. So just a little bit of base pairs that would match each other on opposite strands. Okay, then you reanneal via that microhomology, and what we have is we have flaps that are created, and what you can do is you have an endonuclease coming in, breaking off, okay, cutting those flaps, and then you religate, okay, across. But what happens is each one of those flaps, right, that you've cut off, that contains a bunch of DNA sequence. So that can result in large deletions, which is why alternative end joining is used as a last resort, okay? Because you've lost all this sequence, right? So that can be a bad thing. All right. And finally, we have recombinational repair. So we have homologous recombination, HR, which repairs double-stranded breaks using long homologous sequences, as the name would call out. They use homologous sequences as templates for synthesis. So what is very indicative of HR is these strand invasion events and the holiday junction formation. So the main steps of recombinational repair via homologous recombination is resection. Okay, so same thing, right? We have resection occurring here. So we're cutting away the DNA. Okay, this is now missing. We have strand invasion, which is occurring right here, where this strand, okay, is going into the other DNA duplex of the homologous chromosome. So here's the strand invading. Then you have holiday junction formation. A holiday junction is this right here. Okay, it's also this. Okay. And then we can resolve this either by double strand break repair mechanism or by synthesis-dependent strand annealing. Okay, so either one of these. And we're going to go into more detail what each one of these looks like in the next lecture. But quickly, double strand break repair, we have the formation of our double holiday junctions, we have branch migration, and then we can have either non-crossover or crossover products, whereas synthesis-dependent strand annealing, we just have strand displacement. We don't have any type of cleavage, and we usually result in no crossover product. So I'll leave you with this right here, and this is a table I've put together to help you with each of the repair processes, what type of damage that they will focus on, and the enzymes involved. So remember, mismatch repair is going to focus on replication errors. Okay, and those are going to be our mut S, mut L, mut H, right? Humans are MSH, MLH, PMS. Okay, these are ones that are listed on the previous slide before. Okay, we have base excision repair. Okay, and once again, mismatch repair is MMR. Okay, base excision repair, BER. That is usually used to fix damage caused from ionizing radiation, oxygen radicals, alkylating agents, or spontaneous hydrolysis. And the enzymes involved are our DNA glycosylases and AP endonuclease, and this is for both prokaryotes and eukaryotes. We then go to nucleotide excision repair, okay, NER, and we can use this to fix pyrimidine dimers, bulky adducts from non-IR, so say our UV light, remember our thymidine dimers. This is where the UVR complexes in E. coli come in, or the humans. We have the XP complexes as well as about 25 other proteins. Translesion synthesis, TLS. We do this when we come across an unrepaired pyrimidine dimer, when we come across a site that doesn't have a base, or if we have a bunch of bulky adducts that have gone through one of these MMR, BER, NER processes and haven't been fixed properly. Okay, E. coli. Whether it's E. coli or humans, the Y family polymerases come in because these are the error-prone polymerases that synthesize through these gaps. Remember, this is more of a damage tolerance mechanism than a repair process. Okay, so the Y family polymerases found in E. coli are polymerase 4, polymerase 5. The Y family ones taking part in this in humans or eukaryotes is pol eta, pol iota, and pol kappa. And then we have our recombinational repair, which is taking care of our double strand breaks, and this is going to include our recombinational repair is going to be NHEJ, Alt, EJ, and homologous recombination, okay, HR. And so in E. coli, REC-A, the REC-BCD complex is involved in that. In humans, Q7080 heterodimer, as well as DNA PKCS and Artemis are involved in the human side of that. And we will go over recombinational repair and specifically homologous recombination in the next unit. All right, so that is the end of today's unit. Thank you for joining us at educator.com, and I hope to see you again soon.